Hi everyone. So, after watching the presentation by Dr. Schoenfeld on muscle hypertrophy, I decided to focus on the topic of the role of satellite cells in skeletal muscle adaptation and muscle hypertrophy. So, as a quick review, muscles respond to mechanical load, like lifting, and there's a cascade that causes the muscles to remodel. The three factors that facilitate hypertrophy are tension, muscle damage, and metabolic stress. And there are two types of hypertrophy: contractile hypertrophy and sarcoplasmic hypertrophy. Contractile hypertrophy is when there's an increase in size of muscle fibers because of the addition of sarcomeres in either the parallel or series. Sarcoplasmic hypertrophy is when there's an increase in the number of non-contractile elements like glycosomes and mitochondria. In this kind of hypertrophy, there's a hydration of the muscle cells and the muscles are able to grow. Dr. Schoenfeld's presentation also discussed the increase in protein synthesis through training and the contractile elements that increase after training. One of the mediators of protein synthesis are satellite cells, and that's what I chose to focus on. So, what are satellite cells? They are muscle stem cells that are quiescent. This means they are not active unless they come across a mechanical stimulus, such as a resistance training program. Satellite cells are located between basal lamina and the extracellular matrix and the sarcolemma, or the fiber membrane. Satellite cells have multiple effects on hypertrophy. They can either proliferate, increasing cells through cell division, or differentiate, becoming more specialized to what the muscles need. Satellite cell activation is when mechanical tension can activate the cells and they fuse to the fiber and donate their nuclei. These nuclei make a certain number of proteins, and then the satellite cells donate the nucleus to make the muscle cell more able to synthesize proteins. There can be hypertrophy without satellite cells, and I liked Dr. Schoenfeld's factory analogy he made in his presentation when describing satellite cells. So, at that maximal output, the only way to increase production is to increase the number of factories. So that's the way that the nuclei work to help the muscle cell synthesize more proteins. So I chose the article entitled "The Effects of Heavy Resistance Training and Detraining on Satellite Cells in Human Skeletal Muscles" to see how satellite cells respond to resistance training and the detraining period after a resistance training program. The article discusses how satellite cells can undergo three different pathways: generating new muscle fibers, provide additional myonuclei to the parent fiber, or return to that quiescent status. For the mRNA to increase, which produces those specific proteins through translation, like Dr. Schoenfeld spoke about, there are two pathways. Each myonucleus has to increase the transcription rates of given genes, and satellite cell nuclei have to be incorporated into the parent fiber that's providing more myonuclei. This study was performed with 15 males between the ages of 20 and 32. They were all in good health with no organized sports or heavy resistance training for at least a year prior to the study. The resistance training program was a progressive program with the loading levels monitored continuously and adjusted throughout the entire training period to maintain the muscle loading and avoid plateau. The program was 3 times a week, and it consisted of hack squat, incline leg press, knee extensions, and hamstring curls. And there were 4 to 5 sets of 6 to 12 reps with some upper body exercises thrown in there as well. There was a 3-month detraining period that followed and consisted of everyone returning to their everyday life, so that activity level before entering the study, with no resistance training or exercises performed for 3 months. The investigators performed biopsies on the men in their right vastus lateralis muscle in the leg prior to resistance training, after 30 and 90 days of the program, and then 3, 10, 30, 60, and 90 days of the detraining program. And in the trained state, the biopsies were obtained 24 hours after the last training session. So the investigators cooled the biopsies to about negative 80 degrees Celsius for the analysis, and they had cross sections that were cut at about negative 20 degrees Celsius, and they counted myonuclei and satellite cells on each fiber from these cross sections. And from there, they counted the number of satellite cells per muscle fiber. And this figure just shows you a part of that muscle cross section stained to show the satellite cells in the myonuclei. So the myonuclei are the ones in blue with the full arrow pointing to them, and the satellite cells are the brown ones with just that arrowhead pointing to it. Right. This figure shows the satellite cell number per muscle fiber before training, after 30 days in the resistance training program, after 90 days in the program, and then the detraining program. So the 3, 10, 30, 60, 90. And the little asterisks above the T30, T90, D3, D10, and D60 show that they're significantly different from the pre-training number, and the number sign over the D90 denotes that it's significantly different from the T90 bar. So I thought that this figure was pretty cool because you could see how the satellite cells are increasing, and then how much they really do decrease as the detraining period moves along. So you see it really start to decrease between the 10 and 30 days after the program. Resistance training causes minor fiber damage and leads to mainly hypertrophy. This is compared with the regeneration response of skeletal muscles. So when the muscle fiber dies, it stimulates that inflammatory response and repairs the damaged area with the new sarcolemma. And training is an efficient means to improve the growth capacity of the muscle. In the training period, there are numerous cell divisions, and this leads to that increased number of satellite cells like we saw in the figure before. This enhances protein synthesis after a single exercise session, and it can remain elevated for about 24 hours. So, in conclusion, satellite cells in a mature muscle may have different paths, and they don't necessarily contribute to the formation of new myonuclei. This shows the flexibility of satellite cells after resistance training, and it gives new insights into the effects of training followed by the detraining. The relative changes in the skeletal muscle fiber size can happen without the addition of new myonuclei, and that shows that the myonuclei that already exist can support a certain level of muscle hypertrophy. So I hope you guys enjoyed my presentation, and thank you for listening.